Okay. Hello, everyone. Welcome to Medican Express webinar series. My name is Ian Wan. We are delighted to have you with us for what to be another inspiring sessions. Our speaker today is Professor Johannes Meyer, an expert in field of dermatologic immunotherapy. Professor Meyer currently serves as associate professor at the Department of Dermatology at the University Medical Centers of Mainz. Professor Meyer's academic journey began in Technical University of Munich, where he completed his Bachelor of Science in Biology. After a number of research projects in Germany, France, and the US, he decided to focus on his scientific careers on studying in the intestinal immune systems and parasitic immune responses at the University of Glasgow in the UK, where he completed his PhD in immunology in Professor Simon Milling's groups. Throughout his career, Professor Meyer has been fascinated by how different tissues influence the differentiations and the function of antigen-presenting cells. This interest led him to the Mulligan's Institute of Medicine Research in New Zealand, where he worked with Professor Frank Rancasi, focusing on the dendritic cells and Th2 immune responses in allergic sensitization. In 2021, Professor Meyer founded the DC biology labs at the Philipps University Marburg, focusing on the role of dermal and dendritic cells in both healthy and diseased skins. His groundbreaking work has earned his numerous prestigious awards, including the Werner Müller Prize from the German Society of Immunology and the European Macrophage and Dendritic Cell Society of Young Investigator Research Award. Today, Professor Meyers will share insights into his latest research on early immune mechanisms and the role of skin-specific dendritic cells populations that underlie the common skin diseases. And his contribution to these fields are not only advancing our understandings of immune responses and in the skin, but are also paving the way for novel therapeutic strategies. So without further ado, please join me in welcoming Professor Johannes Meyer to our webinars.
Yeah, thank you very much, Ian, for this nice presentation, and thank you for MCE and Medchem Express for giving me the opportunity today to present some of our latest findings. And so I think we will get started, and I hope you can see my screen. And yeah, it's my great pleasure today to talk about dendritic cells in more detail and how they drive type 2 immunity in the skin, in the intestine, and at different barrier sites, and how we think there is an evolutionary benefit to this development and to these responses, and how we want to use this in making vaccines more effective and also protecting us against infections. I have no conflicts of interest to declare, and the outline of this talk today is to give you a quick introduction into what type 2 immune responses are and how they develop in different barrier tissues. And then the talk is divided into two parts. The first is presenting our earlier work in the initiation of type 2 immunity in the intestine. And then in the second part, comparing these responses and molecular mechanisms to the induction of type 2 immune responses, how we observe it in the skin. And lastly, to conclude what these findings, how these findings can help us in understanding the local immune responses that occur, but also how to use them for therapy and improve vaccination strategies. Just a quick introduction about my current location. So you have heard that my career has taken me to many different countries, not only in Europe and the UK, but also the US and New Zealand. And about 3 years ago, I returned to Germany to start my own junior research group in Marburg. And about 9 months ago, I relocated to Mainz, and I'm currently at the Department of Dermatology at the University Medical Center in Mainz. And you can see the two buildings here, the patient clinic and also our research building that we share with immunology and other departments. And we are involved in a number of research clusters and research programs here locally, which really advance our capabilities. And yeah, I'm really excited to sort of expand our research with this great cluster and group of researchers that are present here locally. The focus in our lab is really understanding the role of dendritic cells, and more specifically type 2 dendritic cells. And our interest really lies in their strong intrinsic plasticity to respond to numerous antigens and drive numerous CD4 helper T responses. And what we're really interested in is to understand how these molecular mechanisms fit to each other, where these different subsets are localized and what role they play in disease. So for example, in the induction of autoimmunity, the induction of allergy, but also in the role of infection and anti-tumor responses. And we use a number of state-of-the-art technologies to do that, to work in vivo and in vitro, but also ex vivo and in patient samples. And we're also currently working on 2D and 3D co-culture models to really assess the complexity and the signaling complexity that those dendritic cells have with numerous non-immune and immune cells. And so now for the introduction of the topic and the initiation of type 2 immune responses. So we mostly know about Th2 immunity from the context of allergy. And allergens can be, are most of the time very inert proteins, for example, from pollen, from mites, from foods, from venoms, that usually by themselves do not drive a normal immune response because they do not have classical TLR agonists. But interestingly, in the context of an allergy sensitization and an allergy response, they do provide, or they do induce, very strong type 2 inflammatory responses, sort of itching, redness, mucus production, and the inflammation of myeloid cells. And this is very peculiar because obviously, if we think about the development of the immune response, it's unlikely that these type 2 immunity has developed against these innate antigens. But actually, these type of immune responses have actually developed to protect and defend us against microbial stimuli, mostly against parasite responses, so large extracellular pathogens that cannot be defeated easily. And there seems to be some sort of miscommunication and immune response that then also initiates this type 2 immune program in the context of allergy. And why this is and how we can sort of stop that is one of the fundamental questions that I have been studying during my career. And so to give a quick introduction into the molecular mechanism and cellular signaling cascade that initiates a type 2 immune response. So most of the time it is associated with tissue damage and the simultaneous uptake of antigens, which some of them have enzymatic properties and they can directly drive or support tissue damage. Some of them do not have just serve as an antigen response, and they need this tissue damage as an intrinsic adjuvant to sort of induce this response. Now, tissue damage in epithelia, so in barrier tissues, is always associated with an alarmin response. So epithelial cells upon tissue damage will produce alarmin cytokines, for example, IL-25, IL-33, and TSLP. And this really activates dendritic cells and drives a Th2 program that also directly drives Th2 immunity. There's also cross communication from these alarmins back to, for example, mucus-producing cells or tuft cells, which then initiate also a non-immune type 2 signaling cascade. And all in all, we sort of have self or foreign antigens or inert antigens that can then, in the context of this inflammation, lead to a type 2 immune response. And this is basically then from the immune perspective what can happen. So you have two arms. In the first arm, there is the adaptive arm where these antigens and signals get integrated and taken up by dendritic cells, and those migrate to the draining lymph nodes where they drive Th2 prime Th2 cells and drive a Th2 response, which is really associated with the type 2 cytokine profile. And you can really see that here with different cytokines, type 2 cytokines, they are really classically involved in this response, for example, IL-4, IL-5, IL-9, IL-13, but also IL-10. And those cytokines recruit and activate additional immune cells such as eosinophils, mast cells, and macrophages that become then M2 polarized and also lead to the maturation of B cells and the development and production of IgE antibodies. And these signals then and their different effector mechanisms then lead to mucus production, local inflammation, and itching, which then sort of drive this type 2 profile, which evolutionary speaking is there to mostly expel parasites and large pathogens that cannot be defeated by our immune cells directly. So the only option is to either encapsulate them by forming granulomas or to expulsing them and getting rid of them, for example, in the intestine through mucus production and flushing them out. There's also a second arm, which is the innate arm of type 2 immunity, and this is a direct activation through alarmin cytokines from the epithelium to innate lymphoid cells and predominantly innate lymphoid cells type 2, which very rapidly and in an antigen unspecific manner also produce large quantities of type 2 cytokines and can recruit and activate the same effector cells. And this is a real, if we look at the global scale, this is a very interesting observation because type 2 immunity really shows a dichotomous distribution around the world. So if we look at asthma, which is sort of a misconcepted type 2 immune response because it's a type 2 inflammation against a non-dangerous antigen, such as pollen or food antigens. We see that actually the developed countries are very strongly suffering from asthma, while the developing countries, so for example, South America, Africa, and parts of Asia, are really affected by the parasite and helminth infection, which also induce type 2 immunity. So there seems to be sort of type 2 immunity all over the world, but it sort of seems to be induced by different antigens. So the sort of evolutionary speaking, parasite-induced type 2 immunity in developing countries, and in more high-income countries, we seem to have an increase in allergies. And there are hypotheses that link these two findings, and this is basically the biodiversity or old friends hypothesis of allergy, which tries to understand this very strong increase in allergy in high-income countries. And it postulates that due to the loss of symbiotic relationships with parasites and bacteria, that were once beneficial and sort of induced a healthy, let's say, type 2 immune response that is actually driving an effective defense. We now have a much more reduced interaction, and we have a lot more virus exposure, a lot less parasite exposure, and that this limited relationship and limited education now leads to this wrong signaling of inert antigens that drive now these allergic type 2 immune responses. And if this is a relationship of the same dendritic cells, so you see dendritic cells being important at both of these early responses and initiation steps, is something that I have been trying to understand during my career. So going a little bit more into details about what those dendritic cells are and how they develop. Dendritic cells develop in the bone marrow continuously from hematopoietic stem cells under the control of FLT3 ligand, where they then develop into common dendritic cell precursors and pre-disease, which then through specific transcription factor programs differentiate into different types of specialized dendritic cell subsets, such as plasmacytoid dendritic cells, which produce large amounts of type 1 interferons in viral immunity, type 1 dendritic cells under the control of IRF8, which are very effective in cross-priming and cross-presentation. So they are very effective in driving CD8 T cell responses, for example, against anti-tumor immunity or against virus-infected cells. And then there are IRF4 dependent type 2 dendritic cells, which drive CD4 T cell priming and basically the entire T helper cell program. And then they are also highly related and in some cases also quite similar myeloid cells, which derive from monocytes and are sort of inflammatory or monocyte-derived dendritic cells, which can also perform antigen-presenting function, especially in the context of inflammation. And there are also other antigen-presenting cells which derive from embryonic precursors and are very similar in their biology to macrophages. And these are most predominantly the Langerhans cells in the epidermis, which do have a very similar phenotype to dendritic cells, but from their development come from embryonic precursors and not from hematopoietic stem cells. Within the intestine, we therefore try to understand which type of these different dendritic cells are important in driving anti-parasite immune responses. And during my PhD, I worked with Andrew MacDonald at the University of Manchester to develop a model where we could understand the response to schistosoma mannsoni X within the intestine, which under physiological conditions get lodged and also encapsulated within the intestinal tissue and induce a strong Th2 response. And we try to understand how this antigen is being processed, taken up, and drives a Th2 response. And so we labeled these antigens fluorescently and injected them into the lamina propria of the intestine of naive C57 black 6 mice. And then within 24 hours, tried to assess where those, which cells were taking up the antigen, the fluorescent antigen, and where they were localized. And so the majority of antigen was actually present within the tissue and the site where we injected the antigen, but there was actually also very specific cell populations that had migrated to the draining lymph nodes and were positive for the antigen. And you can see here that some small proportion of B cells, but a large proportion of dendritic cells were fluorescently labeled with the antigen and were present in the draining lymph nodes. And so we would these cells also induced when we stimulated the lymph node after 5 days of this injection in the intestine. We could also see that there was a strong T cell antigen-driven response that led to the production of interferon gamma, but also type 2 cytokines, so sort of a mixed response, antigen-specific response of Th1 and Th2 cytokines, and that this was actually location specific. So if we injected the X into the intestinal draining lymph nodes, so SMLN, small intestine mesenteric lymph node, showed a very strong response, whereas the colon draining lymph nodes, the CMLNs, did not. But if we injected the X in the colon, the picture was reversed. The colon draining lymph nodes showed a response and the small intestine draining lymph nodes did not, which also showed that in this system, we are looking at a very localized draining effect and a very specific and local immune induction response at this time point. using some state-of-the-art transgenic mice, which actually have a deletion of IRF4 in CD11C positive cells, which actually leads, if you remember the previous diagram, to the deletion or to the defective development of type 2 dendritic cells, while all other subsets of dendritic cells are still there in these mice. We could also show that especially the type 2 immune cytokines were strongly decreased in the absence of of type 2 dendritic cells, whereas the type 1 cytokines were still present and therefore not driven by the type 2 dendritic cells. And this was true in the small intestinal model as well as in the colon egg injection model. Now, when we did some more detailed FACS analysis of the different subsets in the small intestine and in the colon, we could observe that if we use surface markers for CD11B and CD103, that there are actually 4 subsets that can be observed in the small intestine and also in the colon, but that their distribution is quite different. So in the small intestine, you have a very strong double positive population as well as a single positive population, whereas in the colon, this double positive population is much more decreased, and the single positive CD11B positive population is much enhanced. Also in the colon, there are much more of these CD103 single positive dendritic cells, which are actually type 1 dendritic cells, and these 2 upper populations in both cases are type 2 dendritic cells. Now, interestingly, this is also reflected in the draining lymph nodes. So this is in naive mice. And you can also see that in the small intestine, there is a larger proportion of these double positive dendritic cells than in the colon, and that also the type 1 dendritic cells are more pronounced in the colon and colon draining lymph nodes, which means that actually the lymph node and the tissue have a very are mirroring the phenotypes of those dendritic cells, which suggests that there is a continuous migration of all of these subsets to the draining lymph nodes. Now, in the injection model and in the context of schistosoma mensenai egg antigen, we also performed microarray studies to understand the transcriptomic profile and transcriptomic changes of these populations. And we specifically focused on these double positive population and compared it to the single positive population in the context of the small intestine and also in the context of the colon. And what we found was actually that these responses and the activation of these dendritic cells, although by FACS they look quite similar because they fall in the same gate, are actually very distinct. And this suggests that these populations, although they look similar by FACS, actually on a transcriptomic profile and potentially also on a functional level, behave very differently. And we wanted to confirm that by performing another surgical experiment where we basically now took the dendritic cells and injected them. But this time we did not inject them into the intestine, but into the subcapsular sinus of the draining lymph nodes. So basically, fast tracking their migration and just delivering them into the draining lymph node, which is their sort of targeting or tissue. And you can, and this is how that looked under the microscope. So this is sort of microscopic surgery. And you can see that actually when we now delivered sort purified dendritic cell subsets that were stimulated with the antigen and then later collected the lymph node where they were injected and performed restimulation experiments, that this injection of the dendritic cells that were primed was actually sufficient to induce a very effective and very similar immune response to when we injected the antigen in the intestine itself. So basically now we had a very abstract and simplified system where we could really inject specific subsets of primed dendritic cells, and this is what we did. And what we actually found was that if we now took the dendritic cells from the colon, so you remember the CD11B positive DC2s and CD11B negative DC2s, that if we injected those separately into draining lymph nodes of naive mice and then assessed the response, we could actually see that only the CD11B single positive dendritic cells of the colon would initiate a type 2 immune response. Whereas in the intestine, small intestine, the picture was reversed because there the double positive dendritic cells were the major producers of type 2 immunity. And so that again indicates that also functionally these two subsets are very distinct. And so lastly, we wanted to understand of where they are located and also in which tissue under immunologic or physiological conditions they would migrate and drive an intestinal immune response. And if you look at a standard laboratory mouse and you try to identify all the different lymph nodes, you are starting to struggle quite quickly with their small size. And if, for example, you look at the intestine, you sort of see a sausage-like structure, which is sort of a chain of dendritic cell of lymph nodes, but it's very hard to differentiate them individually. And so there is a lymphoproliferative transgenic mouse where all lymph nodes are strongly enhanced about 10 times. And if you look in these mice, you can actually see that there is actually a chain of very individually encapsulated draining lymph nodes. So it's not one big elongated one, but actually individual ones that are just sort of stitched together in a sac. But it suggests that they actually are are individual lymph nodes by themselves, as well as some individual lymph nodes that are loosely associated in other regions of the intestine. And based on these findings and drawings, we were quite interesting to understand which region of the small intestine or the colon were drained by all of these different draining lymph nodes, because we found that this actually makes a difference in the immune response. And so we started to inject very localized dyes and trying to now draw a map of, and you can see here that even the lymphatics and the draining lymph nodes were then discolored within a few seconds, and we could very nicely map then the different regions of the intestine and develop a draining pattern. And what we found was that within this chain of lymph nodes that I had previously shown you, there's actually sort of a mirroring local draining pattern. So the duodenum is drained by the mesenteric lymph node within this chain that is furthest inside the tissue, so closest to the aorta, and also sort of matching this sort of localization. And then as you keep going up to different segments, then further lymph nodes are sort of specifically draining these parts, and these are all within the chain. So even the ileum is sort of the last lymph node in the chain that is being drained. And this lymph node that is separately and a bit larger actually already drains the cecum and the ascending colon, which you can see here. And then the loosely or separately associated lymph nodes that are not part of this chain and sometimes also sit closer to the colon itself actually drain the lower segments of the colon, as you can see here. Now, does this have a functional difference, and that it matters which draining lymph node your antigen is received in, and presumably which types of dendritic cells drive these responses? And yes, it does. So, for example, here in this model, we actually used a live infection of schistosome mansonae, so no injection of X, but the complete infection model. And after 7 weeks is the stage where the X naturally reach the small intestine and drive a Th2 response. And again, we harvested all the different lymph nodes, so the ones draining the small intestine and also the ones draining the colon. And we observed that actually in the colon, in natural infection, there was interferon gamma, so Th1 responses, whereas in the small intestinal draining lymph nodes, we saw that there were antigen present, but that there was a very much increased IL-4 response and very little interferon gamma responses detected, suggesting that they are responding differently, even in a live infection model. And we could also replicate that in a controlled setting using our schistosoma mansonae egg injection model. And here we injected every part of the intestine as shown in the previous slide, and we checked all the draining lymph nodes for the interferon gamma and IL-4 response, and we could confirm also the findings from the live infection model that in the colon and colon draining lymph nodes, the same antigen drives a much higher interferon gamma response compared to the IL-4, whereas in the small intestine, there was a much lower interferon gamma response. And so the ratio of interferon gamma to IL-4 was much reduced in the small intestine draining lymph nodes compared to the colon. And so we took this a step further and not only compared these responses to within the intestine, so comparing small intestine to colon, but also comparing different tissues, so the intestine and the skin. And what was very striking here is that when we injected the same amount of X in a highly controlled scenario, that the immune responses that we observed 5 days later in the skin draining lymph nodes showed that there were much less Th1 responses in the skin draining lymph node compared to the intestinal draining lymph node, and conversely that the Th2 responses, although they were very nicely present in the intestinal draining lymph nodes, were even much more pronounced in the skin. And this was not only true for Schistosoma mentionii X, but also for other parasite antigens. And you can see again that in the skin, the Th1 response against the same amount of antigen was much reduced, whereas the Th2 response was much enhanced. And so we developed a hypothesis based on our findings in the intestine that maybe even beyond the intestinal immune response, there was also another factor in the skin that sort of led to an enhanced type 2 immune response in this tissue. And this then leads me to part 2 of the presentation. But before I get there, I quickly want to summarize the first part, which concluded that type 2 dendritic cells are necessary to prime anti-parasite Th2 responses in the intestine, and that actually this is performed by different subset. So in the small intestine, the CD11b positive, CD103 positive type 2 dendritic cells perform this role, whereas in the colon, this role is performed by the CD11b positive, CD103 negative type 2 dendritic cells, and potentially also these subsets are also using different molecular mechanisms to drive this immune response. Also, I showed you data that different segments of the small intestine and colon are drained by very individual lymph nodes. So the antigen only goes to one lymph node from one specific segment. And that these individual segments also respond differently to the same stimuli. And you might remember this data where I showed you in the colon, the Th1 response was enhanced compared to the IL-4 response in the small intestine. And this very strongly suggests that also within the intestine, there's local immune cell imprinting that differentially regulates responses against the same antigen of the small intestine and of the colon. Now, taking these findings a step further and looking at the different dendritic cell populations in the skin, we started to look at the composition or the subsets that have been published in the literature in barrier tissues. And we actually found that there's a lot of tissue-specific expression patterns, especially in type 2 dendritic cells. So they are highly plastic and can express a number of integrins and other receptors on their surface. And they have the potential at the same time to also induce a very heterogeneous immune response and drive all kinds of T helper cell responses, for example, Th1, Th2, Th9, Th17, and Treg responses, and also others. So there seems to be a very strong phenotypic heterogeneity and also a functional heterogeneity. And we developed two hypotheses to sort of understand how these two might be linked. And in the first hypothesis, we argued that there might be antigen-dependent conditioning of type 2 immune function. So basically, we have a homogeneous profile of type 2 dendritic cells, but all these different subsets have seen different antigens, either artificially or just in a natural context. And this has driven them to change their profile and drive antigen-specific and directed immune responses. The second hypothesis was that actually these subsets are predefined and predetermined, so they develop through signals that are not driven by the antigen, but that these subsets now have a specialized function and only take up or preferentially take up certain antigens or respond to certain antigens in an enhanced manner, and that this then leads to a subset-defined driven differentiation of unique CD4 helper cell responses. And to test these two hypotheses, we first characterized the dendritic cell heterogeneity in different tissues, and we used a high-dimensional full-spectrum flow cytometry approach, where we basically combined all the different markers known in the literature that characterize dendritic cells and dendritic cell subsets, and we performed a high-dimensional analysis of the dendritic cells from within the different draining lymph nodes, so skin, lungs, intestine, and also others that are not shown here. And we could see that actually the profile of those dendritic cells at the steady state was already different. And when we then tried to analyze what sort of markers are the most differentially expressed of these subsets, we could see that in the skin, there in skin draining lymph nodes, there's a unique population of CD11 below type 2 dendritic cells. In the lung, there's a unique expression of CD24 by type 2 dendritic cells. In the intestine, there is the unique population of CD103 positive dendritic cells, which are already talked about previously. And we could show that these tissue-specific profiles were present in the tissue and also in the tissue-draining lymph nodes. So basically that this was not a selective migration of certain populations, but that this actually seemed to be a tissue-specific differentiation that led to the development of these unique subsets, and that this development was also highly conserved because we also found these subsets in male and female healthy mice and also in different strains of mice. And in addition, we again used the type 2 deficient transgenic mouse model to show that all of these subsets were actually dependent on IRF4 and bona fide type 2 dendritic cells. And when we performed transcriptomic analysis of these sorted tissue-specific populations, we actually found that from the transcriptome, they also were quite distinct, and that we could actually identify 6 different clusters, which the most pronounced being the one in the skin, which was defined by the CD11 below DC2s, and the one in the intestine, which were these CD103 positive type 2 dendritic cells. And this is just a PCA plot depicting the same data and again indicating this unique profile of CD11 below DC2s in the skin and CD103 positive DC2s from the intestine. And when we performed transcription factor binding site enrichment analysis to try to understand what were potential upstream regulators that might explain these different transcriptomic profiles, we especially in CD11 below DC2s, so cluster 3B, found that there was a very unique enrichment for potential transcription factor binding sites, and that these were actually grouped into different classes, but which some of them being already involved in specific dendritic cell functions. But to our surprise, there were also a strong enrichment for STAT6. And STAT6 for us was very interesting because STAT6 in T cells drives the Th2 program. And we had observed previously that the skin shows an enhanced Th2 program. And so we were very curious to understand if STAT6 might also regulate some sort of Th2 inducing program in those skin-specific dendritic cells. And so luckily we had STAT6 knockout transgenic mice in the lab, and we performed the same flow cytometric analysis in those mice at the steady state. So here are C57 controls and STAT6 knockout mice from the same animal facility, same age and sex. And you can see that the CD11 below DC2s were completely absent in the skin and also in the skin draining lymph nodes, which you can see summarized here. whereas all the other subsets in the lung, the small intestine, but also within the skin, so you remember I briefly introduced the Langerhans cells at the type 1 dendritic cells, they were unchanged in STAT6 knockout mice. So really only this one specific subset seemed to be dependent on STAT6 signaling. And so we wanted to understand if this was an intrinsic or an extrinsic effect, and we generated mixed bone marrow chimeras. So half of the bone marrow that was then put into an irradiated host was from wild-type bone marrow, and the other half was from STAT6 knockout. And you remember that all dendritic cells develop from bone marrow precursors. So then after several months of reconstitution, we then observed the dendritic cells that had either developed from the wild-type bone marrow or from the STAT6 knockout bone marrow within the same mouse. And we could see that the wild-type derived dendritic cells in the skin and skin-draining lymph nodes had this nice CD11 below DC2 population, but the STAT6 knockout derived bone marrow cells could not develop into the CD11 below DC2s, which indicates that the signal must be within the skin, and it also indicates that the signal then requires the activation of STAT6 to lead to the development of this specific subset. Now, if we look into the signaling cascade of STAT6, there are two activators that can lead to the activation of STAT6, which then drives this transcription program. And this is the type 1 IL-4 receptor, which is from the common gamma chain and the IL-4 receptor alpha. And there is the type 2 receptor complex, which is put together from the IL-4 receptor alpha and the IL-13 RA1. And this second receptor signals both IL-4 and IL-13 and drives the activation of STAT6, whereas the type 1 receptor only can bind IL-4 and drives the activation of STAT6. IL-13 can also bind to the decoy receptor, IL-13 receptor alpha-2, and this can either be soluble and membrane bound, but this receptor does not have a classical signaling domain and does not activate STAT6. So we were left with these two possibilities, either the type 1 or the type 2 receptor. And so we looked at IL-4 versus IL-13 signaling, and we observed that in mice that could not produce or make IL-4 because they were IL-4 knockouts, the CD11b positive or low dendritic cells were still present, whereas in the IL-13 knockout mice, they were absent, which indicated that it must be IL-13 signaling and not IL-4. And we also looked at the IL-4 receptor alpha knockout and IL-13 RA1 knockout mice, and both of them showed a strong decrease in the CD11 below population, which indicated that it's actually a heterodimer of both of these that also, as I indicated, leads to the binding of IL-13 and the activation of STAT6. So we had our molecular pathway that the STAT6 activation is driven by IL-13 through the heterodimer of the type 2 receptor, which is constituted by IL-4 receptor alpha and IL-13 RA1. And so we had proven the necessity that IL-13 is necessary for the differentiation of this unique skin-specific population, but we also wanted to identify if IL-13 was sufficient. And we did this in two manners. We did this in vivo by injecting IL-13 fusion protein in wild-type IL-13 knockout and STAT6 knockout mice, and we could see that in the control scenario, so STAT6, which is downstream of IL-13, we did not see any change or we saw that in both scenarios, CD11 below DC2s were not present. But when we sort of rescued the IL-13 knockout, so not the receptor knockout, but the cytokine knockout with artificial IL-13 fusion protein, this within 4 days would lead to the differentiation of CD11 below DC2s. And the same we could also recapitulate in vitro. So when we added IL-13 to wild-type BMDCs, we would also get the differentiation of CD11 below DC2s in culture, whereas if we did the same with STAT6 knockout or IL-4 receptor alpha knockout, bone marrow cells which are deficient in their signaling of IL-13, we did not see this differentiation. Now, having identified the pathway of how these dendritic cells develop, this skin-specific subset, we wanted to understand their functional importance. And so we devised two in vivo systems. One was a mixed bone marrow chimera, where basically you have mice that have a normal differentiation of dendritic cells in the skin, and also mice that are from STAT6 knockout animals, so they do not have these CD11 below DC2s, and we immunize these mice with different antigens, and we use bacteria, we use parasites, and we use fungi, and we studied the Th2 responses in the skin after the injection of these antigens, and we observed that in the absence of these CD11 below DC2s, we saw no change in the Th1 response against bacterial antigens. When we injected parasite antigens and we did not have the CD11 below DC2s, we saw a very strong decrease in Th2 responses. So they were still there, but they were strongly decreased. And in the context of fungal infection, the absence of CD11 below DC2s led to an increase of Th17 responses. And we also used the second model, which was a Cree-Flox model, selectively targeting the IL-4 receptor on dendritic cells. And again, in this model, you lose the CD11 below DC2s very selectively in the skin. And we observed exactly the same phenotype, no change in Th1 responses, a decrease in Th2 responses, and an increase in Th17 immunity in association with these CD11 below DC2s. Now, we wanted to lastly understand if those dendritic cells were just a murine phenotype or were also found in human skin. And so we performed data mining on published data sets, and we could see that the IL-4, IL-13 signaling pathway and the genes that are linked to it were enriched in all samples that had a skin dendritic cell population in their data sets. So for example, skin versus blood in this first data set showed that for the skin, there is a strong enrichment for IL-4 and IL-13 associated genes compared to the dendritic cells that were sorted from the blood. And here in this data set, they had compared skin dendritic cells versus spleen dendritic cells from healthy donors. And they all, again, in this data set, we observed that in the skin dendritic cells, there was this enrichment for IL-4 and IL-13 pathway. Here again, skin and blood in a second study, and here a last study where they looked at lung versus blood. There's actually much lower enrichment, and you can see this here by these enrichment values, which shows that actually, and this is much more pronounced here in these heat maps, which were generated from the same data sets. So skin versus blood, we see a strong enrichment in these type 2 associated genes in both data sets, but when we look at the lung versus blood data, there does not seem to be an enrichment, which suggests that these signals also play a role or are active in dendritic cells in skin of healthy human donors. And looking at single cell sequencing data, we also observed that type 2 dendritic cells in healthy human tissue also express the IL-4 receptor, IL-13, RA1, and STAT6, which suggests that actually similar profiles might be the case. And we also looked at the homeostatic production of type 2 cytokines, of IL-4 transcripts and IL-13 transcripts. And we could also see that T cells, but also ILCs, are positive for the transcripts and more for IL-13 than IL-4, also in healthy human skin, which might also be the lead to this signaling and to the differentiation of human DC2s. So the summary of the second part of this talk today is that we also found that type 2 dendritic cells in different tissues exhibit unique profiles on a phenotypic level, but also on a functional level, and that in the murine skin, there is a unique population of CD11 below expressing DC2s, which actually develop in response to homeostatically produced IL-13, which is present in the skin at the steady state in mice, but also potentially in humans, and that this subset of CD11 below DC2 DC2s have the intrinsic feature to enhance type 2 immunity and dampen Th17 immunity locally, which might confer an evolutionary benefit to the skin in response to parasite and toxins. And so the sort of construct and model we came up with was that the general response against antigen-dependent responses is that all dendritic cell subsets can perform a non-specialized default response. You remember in our data, we still saw Th2 cytokines and responses being produced, but at a much lower level when these tissue-specific CD11 below DC2s were not there, which means that actually whatever is left over in these mice, in transgenic mice, is actually sufficient to produce a normal or a low Th2 response, but not this enhancement. And so that's why we think there is a general default response where default type 2 dendritic cells can sort of respond to all kinds of antigens and drive a mediocre Th2 or CD4 helper cell response, but that they are also enhancing tissue-specific type 2 populations which perform subset-defined functions, but these enhancing dendritic cells are tissue-specific and have developed based on an evolutionary need. So you don't have all specific subsets in all tissues, but only the ones you really need to sort of help this specific tissue. And so we performed an analysis of many published findings which have looked into specific dendritic cells. And we have found that there are actually numerous subsets which have been described in unique tissues and most pronounced in different barrier tissues. So for example, the nose, the lungs, the intestine, and the skin. And most of them are linked to an evolutionary need. So for example, in the intestine, these specific subsets play a very important role in enhancing tolerance and driving oral tolerance. In the skin, I've already showed you that we have found that they are there to drive enhanced type 2 immune responses. And in the lung, that they are very very strong inducers of driving pro-inflammatory immunity, which might also be linked to the defense against viral responses. And that we postulate that actually these specific subsets are there to help and also evolutionary have evolved to respond to local insults. So for example, within the intestine, we have a huge amount of food and microbiome antigens, which most of them we need to drive tolerance against. So most likely these specific type 2 dendritic cells in the intestine have developed to sort of fill this need of driving food and microbiome tolerance, whereas in the lung and in the nose, we need it is of evolutionary benefit to be able to rapidly respond to viruses and viral infections. So we postulate that in the lung, these dendritic cell subsets are helping us to defend against viral responses. And in the skin, there is the need to defend against parasites, toxin, and mites, which the skin is very frequently exposed to. And these enhanced type 2 immune inducing dendritic cells have potentially evolved to help us in this defense. And so these different subsets and these molecular mechanisms are now for us also novel opportunities to fine-tune vaccine responses and to also fine-tune tissue-specific targeting of antigen delivery, not only in vaccine to help against infection, but also novel vaccines to drive tissue-specific anti-tumor immune responses. And with that, I want to thank you again for the opportunity to present this data. And I hope I could share my fascination and also our findings in the curious role of dendritic cells and tissue-specific dendritic cells in really driving local functions. And lastly, I want to thank all the team members, my supervisors, collaborators, during my different stages of my career, and also our current funding sources, which are helping us move our science forward. Thank you very much. Ian, we cannot hear you. I think you're muted.
Hello. Yeah, thank you, Mayor, for such a great talk. Yes, we have some questions in the chat. Very interesting. Beside the gene enrichment analysis you performed, do you know whether in human that like equivalent DC2 subsets in the skin similar to ones you described in the mice that can be targeted? What is your opinion on the allergen-specific immunotherapy delivered into the skin?
Yeah, that's a very good question and something we are currently working on. The difficulty in studying this, the human dendritic cell subsets, is that you need large amounts of skin biopsies because the dendritic cells are such a rare population, and this is currently something we are working on. So on a transcriptomic level, they seem to be relatively similar, and we could already do that through data mining. But now we're also looking at the proteomic signature, and this is currently work in progress.
Right. Another question. Is there the phenotype and the impairments of activation exhibited by different antigen-presenting cell population during sensitizations essentially the same?
No, it is not. So this is what we have found. So actually the different subsets that we have identified in different barrier tissues in vivo, but also if we sort purify them and in a really controlled in vitro setting stimulate them, we actually see that they respond very differently. And this they actually express a different TLR and receptor profile. So they're even selective in which antigens or which pattern recognition which patterns or danger-associated molecules they can actually respond to. And then also the response and the molecular signaling pathway is also different based on these different signals. So they see different things and they produce different things based on the same antigen. And how this actually happens and how modular this is, or rather how specific, is currently something we are also addressing in the lab by trying to build a mid to high throughput activation screen where we can then have a very large variety of antigens, simple antigens and also complex antigens, so that we can then actually study in a very controlled setting what these different how these different dendritic cell subsets respond to these array of antigens.
Okay, thank you. We have another one. Can those tissue-specific CD2s switch to another types depending on the environmental cues?
Yeah, so here we come to the fundamental question of how plastic are these cells, or rather at what stage of their development is the plasticity stopped? So are they so how can we redifferentiate or further differentiate them or sort of back differentiate them? And so far what we have found is that actually when they have reached the subset specific end stage of differentiation, you cannot turn the clock back. But because, as I mentioned, dendritic cells are continuously produced from the bone marrow, what we actually think is that you can actually differentiate all the new coming dendritic cells, so that there is actually a possibility for therapy by affecting the differentiation of specific subsets locally that are newly arriving, because usually dendritic cells have a turnover rate of 1 to 2 days, so it's very fast. And this actually also gives us an opportunity to actually change or impact this plasticity, and to either change it to get additional benefits locally, or to try to actually inhibit detrimental effects, so where the dendritic cells are doing something wrong.
Okay, great. Talking about therapies, I have this question. What is your opinions on allergy-specific immunotherapy delivered into the skin? And I have a similar question. Do you think it's possible to use like pharmacological interventions for like, for example, like inhibit the status 6 phosphorylation? Is it possible to locally to like modulate the CD2 response?
Yeah, so absolutely, this is therapeutically possible. And what we actually found, so not on the phosphorylation of STAT6, but blocking the IL-4 receptor alpha through dupilumab, which is already treatment in allergy. So biologics therapy, we actually found that the dendritic cells and dendritic cell development is also impacted under dupilumab. And this might actually therefore break the allergen cycle, not only on the Th2 level, but also on the DC2 level. So maybe dupilumab is actually having additional benefits because it also limits the type 2 induction potential of dendritic cells and therefore sort of breaks the vicious cycle in two steps or two areas, the T cell area and the dendritic cell area. But coming back to the immunotherapy and the significance of the immunotherapy being delivered to the skin, if you want to induce, for example, very efficient anti-parasite immunity, this is something we are hypothesizing that, for example, if you vaccinate in the skin, so not in the muscle, but in the skin, you actually get a superior type 2 memory response, which would then ideally protect you against parasite infections. Now, if you want to actually block allergy development and sort of do an immunotherapy, for example, AIT, to sort of limit allergy responses, the skin would actually be the worst place where you could do that, because actually it might even enhance the Th2 response. So there actually, and that's why a lot of AIT is delivered through the mouth, because you want to there target these tolerance-inducing dendritic cells in the intestine, because you want to drive tolerance, you want to drive Treg responses, which then systemically dampen the Th2 response. So there you would not choose the skin because you would get a worse outcome.
Okay.
And I think
Great.
that answers also the two questions from Paul Johansson.
Thank you. Yes, we have another questions. I just gonna pick two now because of the time. I have one question from Paul Jonathan. What is the relative importance of skin and the intestine in sensitizations of food in terms of driving the Th2 immunity to the full allergens?
So there is epidemiological data that actually show that if in babies, if food antigens are first taken up through the skin, that the likelihood of allergen sensitization strongly increases compared to children where these antigens are first taken up through the intestine. So there seems to be a strong correlation that in the skin you get an enhanced likelihood of driving allergy and type 2 associated responses, whereas in the intestine you seem to induce tolerance. So the tissue is very crucial, and we also believe that this is due to these tissue-specific dendritic cell subsets.
Good. Yeah, I'm just gonna have the final questions. Do the different CD2 subsets express different pattern presenting receptors, such as TLRs or CLRs?
Yes, I do. And that's something we're currently working on. And as I mentioned, we're developing this mid to high throughput screening method to actually understand also not only their expression pattern, but also their functional response to these antagonists or agonists.
Okay. Okay. Great. Okay, and thank you very much, Dr. Johannes Meyer, for such a fascinating talk today. And thank you all the audience for coming, attending this webinars. There will be more exciting Medican Express webinars coming soon. Again, Medican Express is committed to provide a platform that enhances the scientific communication and accelerates scientific discovery. So please stay in tune, and I look forward to seeing you all in very soon. So bye now. Thank you.
Great. Thank you again for the opportunity and also for the really interesting discussions afterwards.
Okay, thanks. Bye, everyone.